Hello everyone. We are Prathamesh Rasal, Saurabh Neelkanth, Pradnya Patil, Sanika Zirmile and Omkar Yelkar. We are students of Ashokrao Mane Institute of Pharmaceutical Sciences and Research Save. This video is guided by Mr. A. N. Dhagesar. Subject is Biopharmaceutics and Pharmacokinetics. Question is: discuss the methods used for enhancing bioavailability through enhancement of drug solubility or dissolution rate. Answer is as follows. Methods used for enhancing bioavailability through enhancement of drug solubility or dissolution rate. 1. Micronization. The process involves reducing the size of the solid drug particles to 1 to 10 microns, commonly by spray drying or by use of air attrition methods, fluid energy or jet mill. The process is also called as micro-milling. Examples of drugs whose bioavailability have been increased by micronization include griseofulvin and several steroidal and sulpha drugs. 2. Nanonization. It's a process whereby the drug powder is converted to nanocrystals of sizes 200 to 600 nanometers, example amphotericin B. The main production technologies currently in use to produce drug nanocrystals yield as a product a dispersion of drug nanocrystals in a liquid, typically water, called nanosuspension. There are three basic technologies currently in use to prepare nanoparticles. 1. Pearl milling. 2. Homogenization in water, wet milling as in a colloid mill. 3. Homogenization in non-aqueous media or in water with water-miscible liquids. 3. Supercritical fluid recrystallization, another novel nanosazing and solubilization technology whose application has increased in recent years, is particle size reduction via supercritical fluid (SCF) processes. Supercritical fluids, example carbon dioxide, are fluids whose temperature and pressure are greater than its critical temperature, Tc, and critical pressure, Kp, allowing it to assume the properties of both a liquid and a gas. At near critical temperatures, SCFs are high compressible, allowing moderate changes in pressure to greatly alter the density and mass transport characteristics of a fluid that largely determine its solvent power. Once the drug particles are solubilized within SCF, they may be recrystallized at greatly reduced particle sizes. 4. Use of surfactants. Surfactants are very useful as absorption enhancers and enhance both dissolution rate as well as permeability of drug. They enhance dissolution rate primarily by promoting wetting and penetration of dissolution fluid into the solid drug particles. They are generally used in concentration below their critical micelle concentration values, since above CMC the drug entrapped in micelle structure fails to partition in dissolution fluid. Non-ionic surfactants like polysorbate are widely used. Examples of drugs whose bioavailability have been increased by use of surfactants in formulation include steroids, spironolactone. 5. Use of salt forms. Salts have improved solubility and dissolution characteristics in comparison to the original drug. It is generally accepted that a minimum difference of 3 units between the pKa value of the group and that of its counterion is required to form stable salts. Alkali metal salts of acidic drugs like penicillins and strong acid salts of basic drugs like atropine are more water-soluble than the parent drug. Factors that influence salt selection are physical and chemical properties of the salt, safety of counterion, therapeutic indications and route of administration. Salt formation does have its limitations. It is not feasible to form salts of neutral compounds. It may be difficult to form salts of very weak bases or acids. The salt may be hygroscopic, exhibit polymorphism or has poor processing characteristics. Conversion of salt to free acid or base form of the drug on surface of solid dosage. Form that prevents or retards drug release. Precipitation of unionized drug in the GI milieu that has poor solubility. 6. Use of precipitation inhibitors. A significant increase in free drug concentration above equilibrium solubility results in supersaturation, which can lead to drug precipitation or crystallization. This can be prevented by use of inert polymers such HPMC, PVP, PVA, PEG, etc., which act by one or more of the following mechanisms. Increase the viscosity of crystallization medium, thereby reducing the crystallization rate of drug. Provide a steric barrier to drug molecules and inhibit crystallization through specific intermolecular interactions on growing crystal surfaces. Adsorb onto faces of host crystals, reduce the crystal growth rate of the host and produce smaller crystals. 7. Alteration of pH of the drug microenvironment. This can be achieved in two ways: in situ salt formation and addition of buffers to the formulation. 8. Use of amorphs, anhydrates, solvates and metastable polymorphs depending upon the internal structure of the solid drug. Selection of proper form of drug with greater solubility is important. In general, amorphs are more soluble than metastable polymorphs, anhydrates are more soluble than hydrates, and solvates are more soluble than non-solvates. 9. Solvent deposition. In this method, the poorly aqueous soluble drug such as nifedipine is dissolved in an organic solvent like alcohol and deposited on an inert, hydrophilic solid matrix such as starch or microcrystalline cellulose by evaporation of solvent. 10. Precipitation. In this method, the poorly aqueous soluble drug such as cyclosporin is dissolved in a suitable organic solvent followed by its rapid mixing with a non-solvent to effect precipitation of drug in nano-size particles. The product so prepared is also called as hydrosol. 11. Selective adsorption on insoluble carriers. A highly active adsorbent such as the inorganic clays like bentonite can enhance the dissolution rate of poorly water-soluble drugs such as griseofulvin, indomethacin and prednisone by maintaining the concentration gradient at its maximum. The two reasons suggested for the rapid release of drugs from the surface of clays are the weak physical bonding between the adsorbate and the adsorbent and hydration and swelling of the clay in the aqueous media. 12. Solid solutions. The three means by which the particle size of a drug can be reduced to submicron level are use of solid solutions, use of eutectic mixtures, and use of solid dispersions.